Over the last decade, more than 5,000 papers have been published about TOR, an enzyme inhibited by the drug rapamycin, a drug used experimentally to extend lifespan, but already in use clinically to prevent the rejection of kidney transplants. Patients who received rapamycin due to kidney transplantation had a peculiar side effect, a decrease in cancer incidence. In a set of 15 patients who had biopsy-proven Kaposi sarcoma, a cancer that often affects the skin, within 3 months after starting rapamycin therapy, all cutaneous Kaposi sarcoma lesions had disappeared in all patients. This makes sense, given that TOR functions as a master regulator of cellular growth and proliferation. For example, TOR is upregulated in nearly 100% of advanced human prostate cancers. Maybe that's why dairy consumption has been found to be a major dietary risk factor. We used to think it was just all the hormones in milk, but maybe prostate cancer initiation and progression is also promoted by cow's milk stimulation of TOR. Our understanding of mammalian milk has changed from a quote-unquote simple food to a species-specific endocrine signaling system, which activates TOR, promotes cell growth and proliferation, and suppresses our body's internal house-cleaning mechanisms. Now, normally, milk-mediated TOR stimulation is restricted only to infancy, where we really need that constant signal to our cells to grow and divide. From an evolutionary perspective, it can be concluded that the persistent abuse of the growth-promoting signaling system of bovine milk by drinking milk over our entire lifespan maintains the most important hallmark of cancer biology, sustained proliferative signaling. Grow, grow, grow. TOR appears to play a role in breast cancer, too. Higher TOR expression has been noted in breast cancer tumors and associated with more aggressive disease, and lower survival rate among breast cancer patients. This could explain why women hospitalized for anorexia may end up with only half the risk of breast cancer. Severe caloric restriction in humans may confer protection against invasive breast cancer by suppressing TOR activation. But we don't have to starve ourselves to suppress TOR. Just reducing animal protein intake can attenuate overall TOR activity. Moreover, plants emphasizing plants, especially cruciferous vegetables, not only decrease TOR activation, they also provide natural plant-derived inhibitors of TOR in broccoli, and green tea, and soy, and turmeric, and grapes, along with other fruits and vegetables such as onions, strawberries, blueberries, mangoes, and the skin of cucumbers. Maybe that's why plant-based diets are associated with lower risk for many cancers, the downregulation of TOR. So are we finally on the threshold of being able to fundamentally alter human aging and age-related disease? Only time will tell, but if the pace and direction of recent progress are any indication, the next 5,000 studies on TOR should prove very interesting indeed. Remember this study, in which garlic won out as the #1 anti-cancer vegetable? Well, let's see what it can do. This is what's called a COMET assay, currently the standard technique for the evaluation of DNA damage. What you're looking at is the DNA of a single cell, a normal human breast cell, as visualized under a fluorescence microscope. It's in an electric field trying to pull the negative charged DNA to the right, but our DNA is normally supercoiled tightly together. But you add a carcinogen, like in this case the cooked meat chemical PhIP, that literally breaks up our DNA, you can see the chopped pieces of DNA breaking away and flowing out in kind of like the tail of a comet, which is why they call it the comet tail test. The larger the tail, the more DNA has been broken off into pieces. But if you repeat the experiment, and this time, what if you add the same amount of carcinogen, but Also add in some garlic phytonutrients at the same time. You get some damage, some DNA breakage, but not as much as before. Which kind of garlic would be expected to work the best, garlic or elephant garlic, the so-called garlic for people who don't like garlic? And the answer appears to be garlic. Garlic is best. What about flavonoid phytonutrients found in fruits, vegetables, nuts, and grains? Here are the top 100 sources in the world. Do they have a protective effect on the meat mutagen-induced DNA damage? They took white blood cells from healthy individuals and colon cancer patients and exposed them to increasing doses of two cooked meat carcinogens, IQ, found mostly in fried bacon and baked fish, and FIP, found mostly in fried bacon, fish, and chicken. They used the COMET assay again, measuring how much DNA was broken off in the tail, and as you can see, as the concentration of meat mutagens increases, so does the DNA damage. They then continued to pump in that meat mutagen at the highest level, but started adding some plant phytonutrients. Quercetin, found in foods like apples, red onions, and berries, and rutin, found in like citrus, buckwheat, and asparagus. Even as the highest carcinogen dose continues, adding plant phytonutrients starts to bring the damage down. That happened in both healthy individuals, the solid line, and cancer patients, the dashed line. But I want you to notice something else. Even at a zero concentration of cooked meat chemicals, there was more DNA damage present in the white blood cells circulating in cancer patients. And they didn't have blood cancer, they had colon cancer. Even though the cancer was just in their colon, their whole body was affected by the disease state. Their whole body was under increased oxidative stress, inflicting significantly higher DNA damage. Or maybe the DNA damage came first, and is one of the reasons they have cancer in the first place. Either way, cancer patients experienced less reduction of induced DNA damage, suggesting that higher concentrations of flavonoid would be required to achieve the same protective effect. So cancer and other chronic disease victims need even more fruits and vegetables to reduce the damage done by carcinogens.
I'm here to talk about a really devastating disease. It's created a huge impact on basically everyone's lives. It's killed some of the most well-known celebrities, the most notable Steve Jobs recently. It's called pancreatic cancer. And pancreatic cancer is a devastating disease. Only 5.5% of people who are initially diagnosed will survive after 5 years. And one of the main reasons due to this is that usually once it's diagnosed, the cancer is spread around the body, when the patient has a very small chance of survival. And in order to prevent this late detection, we need a method for early detection, meaning a simple, rapid, sensitive, versatile, and minimally invasive sensor for the early detection of pancreatic cancer. And so there are a variety of proteins that are found in the blood of patients who have pancreatic cancer that are at elevated levels. I chose to say one called mesothelin. Mesothelin, it's highly overexpressed by pancreatic cancer, but not by healthy tissue. But in addition, it's highly overexpressed by these things we call non-invasive precursor lesions, meaning if you can detect this protein, you can detect pancreatic cancer before it becomes invasive, possibly making the survival rate jump above 50% rather than a measly 5.5%. So in order to detect these ultra-low concentrations of mesothelin in your blood, I employed these carbon nanotubes, atom-thick tubes of carbon. And they're really the superheroes of science and material science because they have these broad applications which lead to inexpensive biosensors. In my case, I was looking at how the distance between neighboring carbon nanotubes highly impacts how electricity is transported along them. And so I am really fascinated by carbon nanotubes. I was reading this really interesting paper in my biology class, and all of a sudden we were learning about these new things called antibodies, organic molecules that actually bind specifically to one protein. In my case, it would be to mesothelin, kind of like a lock and key. And so then I thought, in my biology class, I was just sitting there behind my desk looking at this little paper, and I thought, what if I put this antibody in a network of carbon nanotubes, just wildly on a whim? And then it hit me. Amazing. I was very, very happy. My biology teacher wasn't as happy when she found me reading a paper instead of writing an essay on biology class. So then, what I had to have then is a substrate that you can basically support this network of antibodies and carbon nanotubes. And I thought it had to be porous to take up this aqueous solution, but also it had to be inexpensive and readily available. Filter paper, it fulfills all of these. And basically all you have to do is create a uniform dispersion of these antibodies and carbon nanotubes that are just floating around, chilling out. And then you basically just dip coat your sensor, making it really easy to produce. And so here's a visual depiction of the sensor being created. And how you test it, basically, it only requires 1/6 of a drop. And you just put it on, and then you just measure the change in electrical potential, just an electric parameter. So then, in order to detect pancreatic cancer from the detection of this protein, basically what you have to do is you have to establish what we call a cutoff level, where anything above that level of mesothelin is considered positive, anything below we consider negative. And so usually it's 10 nanograms per ml for mesothelin, so that's what I chose. And so then I ran in vitro study just to make sure it was working pretty well, and it worked spectacularly, actually. And then we went into actually testing human samples. We had patients who had pancreatic cancer and then also the non-invasive precursor lesions. You can see they all fall in this range of 18 to 28 nanograms per ml. That's well above the 10 nanogram per ml cutoff. All of these would be found positive by my sensor, meaning that it had 100% sensitivity towards pancreatic cancer. Then we had patients who did not have pancreatic cancer. They are either perfectly healthy and skipping around, or they had chronic pancreatitis, meaning an inflammation of the pancreas, the disease most often mistaken in diagnosis of pancreatic cancer. All of these you can see are below 10 nanograms per ml, meaning that they would all be found negative by my sensor, meaning that it had 100% selectivity towards pancreatic cancer. So it was giving near perfect diagnosis of pancreatic cancer, thus revolutionizing how we look at cancer diagnosis. Thanks. So then what we were looking at another application of the sensor. We wanted to see if it could effectively look at how well a treatment was doing on a patient. What we had is 5 chemotherapeutic drugs were given to patients over 5 weeks, and you can see for each of the 5 chemotherapeutic drugs, there were distinctive drops, so it could reliably monitor how effective a cancer treatment was. And so what happens is this sensor, it's really revolutionizing how we look at cancer treatment and diagnosis, because compared to the current gold standard of protein detection in cancer, it's 168 times faster, over 26,000 times less expensive at 3 cents, and over 400 times more sensitive. And also, it has limitless applications. You switch out the antibody, and you can detect an entirely new protein, meaning you can look at other forms of cancer, but also you can look at cancer treatments effectively. You could look at cancer drug resistance, because these are caused by unique mutations in the DNA, producing a unique protein, which you can pick up with my sensor. Also, you can look at the effectiveness of a cancer treatment, meaning that we are revolutionizing how we look at cancer treatment, personalizing the treatment at a patient, so you don't have that conventional guess and check method. Also could look at other forms of disease, such as in food and water safety, E. coli, salmonella, rotavirus, and then also you could look at other blood pathogens such as AIDS, STDs, and all of those. And so it could operate in any setting, such as a regular checkup, but also in developing nations, revolutionizing and possibly saving thousands of lives in cancer diagnosis, treatment, and the biomedical field. Thanks.